Well, it is the last pre-recorded class for Elementary. This is Beans in a Bag. And I tell you what, I may have saved one of the best activities for last. And so in this session, I'm going to show you the Beans in a Bag inquiry lab and how you can easily just adjust lab activities to engage students in inquiry practices. And so this one leads to being a guided inquiry and also an open inquiry as well. And so here we go. So before we begin, allow me to give a little bit of instruction behind all the different stages of the germination and all of the parts that you're going to want to identify with students of your beans while they're germinating. And so first of all, the bean has a seed coat which protects the cotyledons or the embryos and things inside of the seed. And so whenever you wet the seed coat, water absorbs through it and it activates the germination process. Okay, and so germination is the act of changing from seed to plant. And so the seed starts to split into two cotyledons, and the radicle of the embryo will begin to emerge. And so when you take a look here, this long structure that's kind of bent inside of the bead starts poking out, and it'll be the first thing that you observe growing whenever you do this activity. And so it'll be pretty exciting seeing that. And then that will continue, the embryo will continue emerging out until the hypocotyl, which is a little bit of a green color where the stem forms, that will become visible after some days of observing and waiting. And gravity will pull the root of this, so where the radicle was will become the root side, and root hairs will begin growing off of this. Whereas the hypocotyl will really be the part of the stem that's, connecting the soil with the air above, and it will grow up from that soil, and the cotyledons within the bead cell will push up above the surface and turn into the leaves of this bean plant. And so forgot to show you some of my images. So here's like day 1 or 2 after I had dampened my bean, my pinto bean. You can see the radicle starting to emerge there. And then about 3 days after, you could start seeing the rest of the embryo growing out. So it's still inside of the bag. You can see the little water droplets that are adhering to the plastic bag, and the paper towel is behind it. And then here it was about 4 or 5 days, and you could see the radicle starting to move down. And so this part, the seed coat was still on, and so the cotyledons, which are these big embryos inside this, the cotyledons for the bean actually hold all the nutrients that it needs to grow until the first leaves sprout. And so all that food source is in there, and that's why it's being protected by the seed coat. And then it just turns into the plant. And so it's really a beautiful and exciting thing to watch. And it's a great process that students can do drawings for observations for, and also learn how to write for sequencing purposes. So that this is a sequence that they can actually start writing about in scientific purposes. So this is what they'll look like. And so notice in this video there are a bunch of beans. I think there might actually be a little bit too many. I would suggest putting 3 beans per bag so that students can have 3 different samples to analyze, but also not too many so that the roots don't start getting tangled up whenever you try to pull them out. And so you can actually see here, this bean plant has shed its seed coat already, and the cotyledons are exposed here, and you can see the little leaves, the 2 leaves emerging, 1 out of each cotyledon. So the cotyledons will turn into the first leaves for this plant, and then the I believe it's called the epicotyl will start growing on top, and another stem starts coming out, and so the plant just gets taller and taller. And so then the leaves are responsible for pulling in carbon dioxide and sunlight to actually do photosynthesis to allow for the plant to grow and live. And so wow, and so at this point, whenever the leaves have actually started emerging, that means it's ready. You're ready to transplant it. So you're going to carefully take them out of the bag, and you can put them in the soil and with a pot, and that's going to allow you to start opening up this activity into the inquiry process, where students can decide the procedure that they're going to do to grow their plant. So they can identify which variable they want to test. Maybe they want to test the amount of water that they water the plant each day. Maybe they want to test the amount of sunlight. Maybe they want to test the color of light. And so they're going to come up with procedures to test this. But we'll get to that in a bit. First, I want to show you guys some of these amazing videos. And I mean, they're just, these time-lapse videos of these bean plants growing are just so gorgeous. Like, I had to put two in. I couldn't pick just one because they're so amazing. So we'll start with this one. Here they're doing it in a jar instead of plastic bags. The plastic bags allow you to tape it to a window in your classroom, so it's not taking up space in your classroom until later. So I like this video because I like this video because it shows what the root hairs would do if they weren't in soil, and I thought that was pretty neat. So this is what it is when it's in soil. This is a cross section, and what I like about this video, pay special attention to the cotyledons and how they transform into leaves, and then help the plant grow. And also you can watch kind of like how parts of the plant that are no longer needed also kind of fall off. And the music's cool too. Those cotyledons are just coming up, right? Like, so this is the bean. This was the bean. Makes me think of Jack and the Beanstalk a little bit. So watch there, the seed coat, it falls off. Here are the cotyledons. So they're no longer needed either. They're eventually going to fall off now that the leaves are going to photosynthesis. There's that epicotyl growing up. Notice that the cotyledons have completely fallen off of the plant at this point. could just watch that over and over a couple of times. That is so cool. All right. So I also want to show you corn seed growing, because this is a monocot, right? It's only got one cotyledon, and so you can see how it grows a little bit differently. So it's only going to have a single cotyledon that comes out to form the plant. Is left behind this time, right? So it's still holding the embryo that's providing the food for the plant until this first leaf comes out of the cotyledon. Whereas with the bean seed, the embryos came up and were part of the cotyledon. I guess it's going in reverse now. Just so cool. Okay, so this could be open inquiry if you provide your students with just different seeds, right? Different beans and seeds. So you could do lima beans, kidney beans, pinto beans, corn seeds, rice seeds, and have them decide, you know, what they're going to test, and just leave it open to them to come up with questions about the seeds. And so they could observe the difference between the dicots and the monocots through their investigations. And so, yeah, some of these are just cool, really cool to learn about. And so we're going to, wanted to start this video, but I guess in the background is this image of the differences between dicots and monocots, not just within the seed, but also the plant. And so this guy's going to show you the difference between the seeds of dicots and monocots. The monocot is on the bottom here, the dicotyledon is on the top, and so you can see inside the two cotyledons are there. And here there's only one cotyledon that comes out of the plant. And so the cotyledons here in the lima bean are actually the whole endosperm. So this is a little bit pictured differently than what he's going to show you. And then down here, the endosperm here, this stays within the seed, right? Like we saw in the corn seed. This stays, and so the food remains here in the seed. And what comes out is this side with the cotyledon that produces the first leaves. And then the networks of veins on the leaves are different. Monocots have these straight line type veins, parallel veins, whereas dicotyledons have a network of veins that branch off from one another. And then the vascular rings, so where the xylem and phloem are, where the water comes up through capillary action, those form a ring shape in the stem of the plant, whereas in a monocot, they're just kind of dispersed just throughout in random ways rather than being in a ring shape. And then for a monocot, you get 3 flowers whenever you have a flowering plant, whereas your dicots do it in groups of 4 or 5 usually. And so those are the properties between the dicot plants and the monocot plants. Let's take a look at this seed dissection. This is pretty neat.
Okay, today we're going to be doing a seed dissection, and I've got a corn seed, which is going to be our monocot seed, and I've got two dicot seeds. I've got a kidney bean seed and a lima bean seed. Those are going to be our dicot seeds. We're going to go ahead and get started with the corn seed first. And what I'm going to do is I'm going to look over the outside of the corn seed, and I'm going to look for this white sort of apron-like structure. You can see it right here, this white apron-like structure. And this is going to be the embryo, and we want to look for that, because if we cut the seed in the wrong plane, we're not going to see the structures that we need to see. So let's go ahead, we're going to cut this white apron-like structure right here. And I'm going to take my scalpel and I'm going to cut that. Be very careful when you cut this. You do need to hold the seed, but be very careful how you actually cut this seed. So I'm going to cut right in the center of that white apron or embryo. And you can see I cut that right in half. And the next thing I'm going to do is I'm going to put a little bit of Lugol's iodine on it, just a very light coating to sort of bring out some of the structures. So a very light coating. And I'll put just a little bit on the other half of this. I'm going to let that soak in for just a second. And there we go. So let's go over the parts of this monocot seed. And this is going to be a little difficult, but the outer part, you can see that there's an outer tough layer. I'm going to get the dissecting needle underneath it. But there's this outer tough layer. You can see I'm actually teasing back a little piece of it here. There it is right there. That's going to be the seed coat, and that is a protective covering, mainly to protect the seed against digestion when other animals eat it. But let's take a look at some of the other structures here. The really dark material right there, that's going to be endosperm. It stains deeply black because it's got a lot of sugar in it. So that deeply black staining structure is going to be the endosperm. The lighter, more gray staining region right here is going to be the cotyledon. That's another food source for the developing embryo. And this very light area right in here, almost like a sliver, this very light area is going to be the embryo. That's going to be our developing embryo right there. And those are the structures we want to see. Seed coat, seed coat, endosperm, cotyledon, and embryo. Now, let's go ahead and move on to the dicot seed. And I'm going to start with the lima bean first. Now, this has been soaking for about 8 hours or so it comes off pretty easy. You can see that just by moving my fingers that the seed coat is literally falling right off. So that's the seed coat. I'm going to put that aside, and then very gently, you can see if I point right here, you can see a little bit of the embryo sticking out right there. And we don't want to damage that. We want to try to keep that intact. And I'm going to just ever so gently, just give it a little pinch, just a very small pinch. This is a dicot, so it's going to split in two normally. And as I open that up, you can see the two halves of the bean seed. And what you're looking at right here, this is all going to be cotyledon, food for the developing embryo, all cotyledon. And then you can see this structure right here. This is going to be our embryo. So what we can do sometimes, if it's not obvious right away, is I can put just a very small amount of iodine on here. And then I'm just going to pat this. And you can see this white area right here. This is the part of the embryo right here. This is going to be the epicotyl or plumule. And then I like to divide this into thirds. So that would be the top third. The middle third right here would be the hypocotyl. And the lower third right here would be the radicle, which will develop into our root. And those are the parts of the bean seed that you want to see. Now lastly, I have a kidney bean here, and you can see if I pinch it a little bit, it's not going to come apart. So what I'm going to do is I'm going to take the very tip of the scalpel, hold it like a pencil, actually going to put it in the tray, and just with the very tip of the scalpel, I'm just going to cut through that seed coat. I'm not going to go into the seed hardly at all. And that should be enough to peel away the seed coat. And as you can see, I'm starting to peel it away ever so gently. I'm going to peel that away. That's all the seed coat. And I'm taking care not to squeeze it or pinch it too hard. I don't want to damage the embryo. And you can see the embryo sticking out right there just a little bit. Now I'm going to give it just a little pinch so I can pull this apart. There we go. It should just break in two automatically because it is a dicot seed. Let me move some of these other parts out of the way. I'm going to dab this just a little bit. I'm going to put a little bit of iodine on here so to bring out the color. Just a very small amount. And I'm going to soak it up just a little bit there. And if we look in here, you can see the embryo right here. Here's the embryo. And then you can see the top part of that embryo right there, that epicotyl or plumule. And then the middle third is going to be the hypocotyl. And then the lower third is going to be the radicle. And again, all of this other area right here is going to be our cotyledon, all cotyledon, all cotyledon, which is going to be our future food for our developing embryo. And that's going to be it for the bean seed dissection.
Pretty neat. And so with my experiment that I had done to prepare some of the pictures for you guys, did have some problems along the way. I had a seed fall and it broke the embryo. And so I'm going to pull out my seeds here and I'll show them to you. So you can kind of see here, these are my embryo, and notice that, or these are my cotyledons, sorry, notice that there's no embryo on it, and that's because it fell and broke while it was on its way out of the seed. And so the seed coat is still here, it kind of fell off as well. And then this is the little embryo that broke off when it fell. And so that's why it's definitely a good idea to have at least 3 seeds in there so that way if something like that happens to one of your kids' seeds, they'll be able to do the rest of the experiment. And so here's my pinto bean, and you can see how that radicle was formed, and then the hairs are coming off of it, and then you can also tell this little green side now is it's the hypocotyl. I'm really tempted to like break it open, but the seed is a lot harder than the seed coat is a lot harder than the lime. I mean, oh, it's working. Okay, so I'm peeling it back now. We're gonna reveal what this looks like, which to no surprise is going to look like the other dicot bean seeds that we've seen. But I don't need any iodine solution to show this, right? This is a very visual thing. So it might not be a bad idea to do a little dissection, kind of like I'm doing right now with a couple of seeds. that haven't fully, aren't going to fully finish the process. So there's I split them apart. They're both attached to the hypocotyledon, right? So it's providing food source to the whole plant, both sides. Oh, I just broke it off. And then you can actually see inside of here, there are two little leaves. And it's kind of hard to see in the video, but there are two leaves already there, kind of in their baby form. I don't know if I can angle it a little bit differently to show you. There we go. How cool is that? Okay, so on to inquiry now. Remember, guided inquiry is where the teacher provides the investigatable question, and open inquiry is where students come up with their question. And so I can see this being an opportunity to guide students into developing their own question. Like you present them with some of the materials, like, well, here are our now grown bean plants, and they're just going to continue to grow. What kind of science questions can you come up with? And students can decide some questions, and from there they can develop a procedure to test those questions. And so in the guided inquiry, you could provide them with an investigatable question. You can actually ask them to do something particular, like how does the amount of water affect the height of a bean plant? Or if you're growing seeds, corn seeds, the height of a corn plant over a certain number of days. And so you can provide that question to them, or you can let them decide what their independent variables are going to be and let them develop the procedure themselves. And again, you can also just start by giving them random seeds and show them how one can grow in a bag whenever it's wrapped in a damp paper towel. Actually, you don't fully want to dampen the paper towel, or you do want to dampen the paper towel, but you don't want to fully wrap the bean, so that way you can see it. You want to leave it on one side of the bag against the bag, and then the paper towel, the damp paper towel, can be like leaning up against it. You can show them how that works, and then say, well, here's some seeds. Come up with an experiment. So guided inquiry, here are some possible ideas that I had for investigatable questions. So the first one is, how does the amount of water given to a bean plant affect its growth? Another question would be, how does the color of light affect the bean's growth? So you might get some grow lights and use particular colored grow lights, or you can wrap regular lights with like cellophane or saran wrap that are different colors. How does the depth of seed burial affect the growth of a lima bean? So just planting them different deepnesses into different pots could be a particular investigatable question. How does the temperature of water affect bean plant growth? Might be an interesting investigation. How does the amount of light affect the bean's growth? And how do fertilizer beads affect the growth of a bean plant? Could be another thing. So different types of fertilizers. So you would want to guide students to their dependent variable. If you're providing them with the investigatable question, if they're struggling with coming up with a dependent variable on their own, then you're going to have to ask them questions to guide them there. Don't just tell them, but ask them good questions to guide them in that direction. So some example questions would be, well, how will you determine if such and such amount of water helped or hurt the plant? Or another question, well, what will you do each day to observe and measure the plant? You can use another example of somebody in the class that's come up with an idea. They're like, Sally plans to measure the thickness of a stem of her plant with a tape measure. Do you have a unique idea in mind for how you're going to measure your plant? And so remember, a guided inquiry is when students develop the procedure themselves. So challenge them to earn your approval for their experiment by writing the steps out for doing the experiment, and they will use this set of directions also throughout the experiment. They're going to come back to it and remember exactly what it is that they need to do every day. And so you may want to guide them by providing a sheet that has them identify on the sheet, this is my independent variable, these are my controlled variables, and then a list where I'm going to write, this is what I'm going to do every day to measure this particular thing that I'm testing. And so this is a great way to have students writing out a plan for their experimenting. And so for open inquiry, remember, this is where the students are coming up with their own question, but you can actually help them come up with a question by giving them a particular topic, like we're growing beans. So what kind of experiment can you create from these beans that we're going to grow? Students should have already been trained, though, on those elements of designing experiment, like I keep mentioning, right? Don't just expect them to jump in and be able to develop their own experiment if they haven't practiced those integrated science process skills and their basic science process skills. They're not going to know what to do. And so they should have already practiced those things, formulating hypotheses, controlling variables, interpreting data, experimenting, etc. And so what observable phenomenon could you investigate? Might be a great way to ask them a question. Well, what can you do? Do you have an interest in anything that might be able to be used as a science experiment, such as gardening, skateboarding, or coloring? You know, those are topics that are also outside of the open inquiry phase. But let's do some research on this topic also. I wonder what we can learn by reading that also will help us come up with something that we can test. So maybe do a little bit of research about bean plants and help them think about, you know, things that they're going to be looking for as well. Instead of telling them, let them do a little bit of research. And so students then are going to write out their practice procedure, and you're either going to approve it or deny it also in the open inquiry phase. Okay, so we have spent the day today talking about different levels of inquiry. We had a video on the sun, moon, and stars where we focused on confirmation and structured inquiry. And then this particular video is a good guided inquiry, which also could be easily adapted into your students' first open inquiry process that allow them to start developing their own open inquiries later on in your class. So now I want you to reflect on the different levels of inquiry. And so the last journal entry for the course, just reflect on these three things. Which type of inquiry activity are you interested in using the most and why? And number two, does the inquiry model you selected lend itself to the math integration that you discussed in your post for number 6? And so why does it or why not? Can you turn your math practice into an inquiry lab in some way? And then number 3, can you include the reading strategies from post number 8 into the inquiry model? Why or why not? And so how will those things look, or what ideas do you have if you're going to try to integrate them? And it's not required that you have something. I'd just rather you explain why not, if not. So make sure you fully answer those questions. And also come back and check out these links, guys. There are some really great websites that I use to create the information that I shared with you in this presentation. So thank you guys so much for being a part of this course. Make sure that you also take the post survey after you finish the class. All the day 5 activities. So after the teleconference, you can do it. But don't forget that that's also something that I need you to do so that we can use the information to make our class a little bit better in the future. Okay, so again, if you guys have any questions, email me, and I look forward to continue working with you. Just because this course is over doesn't mean that you can't reach out to me with your questions, and that I won't be able to give you some more ideas and help you through your process of growing as a teacher. And in turn, it's going to help me become a better teacher as well. So I would greatly appreciate us collaborating. Hey guys, have a great day.